Greetings and welcome once again to Trading Grades Online School. Today, I'll be taking you through Mumias Sub-County Joint Evaluation Test, Biology Paper 1. Please, if you are new to this channel, find your time to like our video, share our video, comment, and most importantly, subscribe to our YouTube channel, Trading Grades Online School, for you to be notified whenever we drop any video of this kind. Welcome. Question number 1. Distinguish between magnification and microscopy. These are terms used under the topic the cell. We are going to define or differentiate the two terms, distinguish the two terms magnification and microscopy. So magnification, it is the process of making an image of an object to appear large as compared to the actual size. And then we have microscopy. Microscopy is basically the science of using a microscope. So we talk about microscopy being the use of a microscope to view and study objects that we are not in a position to observe them with our naked eyes. So how do we distinguish the two? So I'm going to use the simplest terms as possible for you to understand. So we are going to define magnification. So magnification is the process of making an image of an object, to appear larger than its actual size. To appear larger than its actual size. And because we are distinguishing, then we say while microscopy is the use of a microscope, or is the process of using a microscope, is the use of a microscope, to view and study objects that are too small to be seen, and study objects, which are too small to be seen with naked or unaided eyes. So basically, that is the difference between magnification and microscopy. So giving the correct difference, you are supposed to get 2 marks. Remember, stating one of the statements being correct and the other one being wrong, then the whole question is, or the whole answer becomes wrong, simply because we want the difference between the two. So if you score one and you get the other one wrong, then it means you have not given a difference, or you have not distinguished between the two terms. So I want us now to move to part B of question number 1. List any 3 adaptations of the root hair cells to their functions. So we know the root hair cell is one of the specialized cells in plants. And this root hair cell is special in different ways. What is this that is special with this particular cell? This is what we call a root hair cell. You can see we have an extension here. We have an extension. We have a new vacuole, a sub-vacuole. We have cell wall, and it contains all other parts of a cell except chloroplast. The root hair cells do not have a chloroplast. Now, let's look at some of the adaptations of this particular cell that makes it carry out the process of absorbing water and mineral salts. So because the root hair cells for absorbing water and mineral salts, let's look at some of the adaptations. One of the adaptations is that this particular cell have a long and thin extension that provide a large surface area for absorption of water and mineral salts. So we can put that statement down and say they have a long and thin extension to provide large surface area for absorption of water and mineral salts. For absorption of water and mineral salts. Another adaptation is that they have thin cell wall that makes them to allow water to enter the cell easily. So we say they have thin cell wall to reduce the distance covered by water and mineral salts. Remember the function of root hair cells to absorb water and mineral salts. Then we have another point is on the numerous number of mitochondria within the cell, and this is to provide energy for active absorption of mineral salts. So they have many mitochondria or numerous. They have numerous mitochondria. Remember, we have one is mitochondrial, many are mitochondria. They have many mitochondria to provide energy, for active absorption of mineral salts. For active absorption of mineral salts. So stating 2 or 3 points, then you score 3 marks. We still have more adaptations. The vacuole contains, we have a very large vacuole, cell vacuole, that makes it to maintain a low water potential, which allows water to enter by osmosis. We also have the lack chloroplast. They do not have a chloroplast to create more room for the large cell vacuole. We also have these ones. These roots are numerous. These root hair cells are very many to increase surface area for absorption of water and mineral salts. Let's move to the next question, part 2, question 2. Why would you give an athlete glucose and not sucrose after a race? We need to understand that under the categories of carbohydrates, we have three major categories of carbohydrates. And these three major types of carbohydrates, we have the monosaccharides, we have disaccharides, and we have polysaccharides. Monosaccharides are the simplest form of carbohydrates, meaning whenever you take any meal that contains carbohydrates in form of polysaccharides or disaccharides, they have to be broken down. They have to undergo the process of digestion so that they are broken down to a form that is easily absorbed into the bloodstream. And some of the examples of monosaccharides, we have glucose. We also have galactose. We also have fructose. On the side of disaccharides, we have sucrose, which is formed from combination of we have fructose and glucose. We also have lactose formed from galactose and glucose. We also have maltose that is formed from two glucose molecules. So those are disaccharides, and they need to undergo a further process of digestion for a simpler form to be obtained. Polysaccharides, we know we have starch, cellulose, glycogen. These are some of the examples of starch or the polysaccharides, and therefore they also need to undergo a process of digestion for them to be in a position to be absorbed. So they are a bit less soluble or insoluble in either blood or water. So why is it that this athlete was given glucose? Glucose is a bit more soluble in blood, so it is easier for glucose to be absorbed than sucrose. Sucrose is a disaccharide, while glucose is a monosaccharide. So it is easier for glucose to be absorbed than the sucrose, which is a disaccharide. So we write and say glucose is highly soluble in water or blood. So in this case, it's blood. So we say glucose is highly soluble in blood than sucrose. And hence faster transported to the respiratory tissues or cells, and hence faster transported to the respiratory cells or tissues. So that statement is enough for 1 mark. Glucose is highly soluble because it is in its simple form, so it is easily absorbed into the blood. Therefore, this statement was enough to give you 1 mark. Let's move to part B of this question. What happens to lactic acid after oxygen debt recovery? What happens to lactic acid after oxygen debt recovery? We know during the process of respiration, both in aerobic and anaerobic. In aerobic respiration, we know we have two phases. We have the glycolysis and we have the Krebs cycle. During glycolysis, glucose molecule is broken down to pyruvic acid, and in cases of less supply of oxygen, pyruvic acid in animals is broken down to lactic acid. Pyruvic acid in animals is broken down to lactic acid. And what happens? This lactic acid then accumulates in the body muscles. So we have said in animals, pyruvic acid is broken down to lactic acid, while in plants, this pyruvic acid is broken down to ethanol. We also have carbon dioxide and some water. So this is in plants. And we are in our question, in our case here, we are looking at this lactic acid that accumulates in the body muscles. It has to be removed, or it has to be broken down when the oxygen debt is paid. So this oxygen, we know we have what we call the oxygen debt, the extra amount of oxygen that is required to break or to get rid of the lactic acid that accumulates in the body muscle when that supply of oxygen was less than demand. So we are being told what happened to that lactic acid after the oxygen that was needed has been attained, or that debt has been paid. What happens to that, or to that particular lactic acid? So we have the lactic acid being broken down or being oxidized into carbon dioxide and water. The lactic acid can be broken down into carbon dioxide, water, and energy within those particular muscles or those tissues. So we can say it is broken down into carbon dioxide, water, and energy is produced. Another activity that takes place, or another thing that happens to the lactic acid, it is transported to the liver and then converted to either glucose or glycogen or fats for storage. Is transported to the liver and then it's converted into glucose, glycogen, or fats for storage. So those are some of the processes that occur to this particular lactic acid that accumulates in the body muscle. Let's look at question number 3. What is gene mutation as used in the topic genetics? Gene mutation means what? We know we have the processes that take place in on the gene structure, or the structure of that gene, making it to change its form. So these factors may be environmental, or they may get initiated from the body due to the process of gamete formation. So what is this gene mutation? Gene mutation, we refer to them as spontaneous changes, or is a spontaneous change, is a spontaneous change that occurs in the genetic makeup of an organism. So we have seen types of gene mutation, like we have deletion, we have inversion. So these changes that take place in the genetic makeup of an organism is what we call gene mutation. So stating this, gives you 1 mark. Let's move to part B of this question. State 2 disorders in human beings caused by gene mutations. We have those disorders that affect human beings, and they are caused or they are due to gene mutations. And we have several of them, including sickle cell anemia, albinism. You can also talk about hemophilia, color blindness. All those are disorders in human beings that are caused by gene mutations. So we have sickle cell anemia. We have sickle cell anemia. And sickle cell anemia is caused by a mutation of a gene that produces hemoglobin in the red blood cells, or the red blood cells, they are sickle shaped. They make the red blood cells to appear sickle shaped. So that is what we call the sickle cell anemia. So it becomes a problem for these particular cells to transport hemoglobin, or to pack enough hemoglobin for oxygen transportation, and that's how it becomes a condition. Then we have albinism. Albinism, this is a disorder that is due to a mutation of a gene that is involved in the melanin production. So you find that that gene is not in the capacity to produce enough melanin that gives the skin its color. Then we have hemophilia or hemophilia. Hemophilia is a condition or disorder that is caused due to a mutation of a gene that responds to blood clotting. So you realize that blood is continuously, in case of an injury, the victim continuously bleeds. So that gene disorder or that mutation that occurs leads to a disorder that we call hemophilia, excessive bleeding. Then we have another disorder in human beings that we call color blindness. Color blindness. Color blindness is due to a gene mutation for light sensitive pigments in the eyes. So that light sensitive pigments in the eyes are not able to develop fully because of this mutation that occurs leading to color blindness. So stating only 2 here was enough to give you 2 marks. Let's move to part C of this question. What name is given to the factors in the environment that encourage or speed up a mutation? Those factors in the environment that lead to cases of mutation, like we have heard of certain atomic bombs, certain heavy metals that lead to cases of mutation. So the name that is given to those factors, we call them mutagens, or sometimes we call them mutagenic agents. So we call them mutagens. We call them mutagens. Number 4, state the importance of the following in living organisms. This is one of the questions that is wrongly interpreted by most of the students. We are being taught to state the importance of these characteristics of living organisms. We have several characteristics, including the ones that we are given here: nutrition and excretion. We also have gaseous exchange, respiration, irritability and sensitivity, growth and development, and other characteristics. But in this case, we are being taught to state the importance of these particular characteristics to the living organisms. So we are going to state how they help these organisms. So nutrition, we know it's the process by which organisms obtain and utilize nutrients. So when they obtain these nutrients, how do they help the organism? So So they provide living organisms with nutrients that are oxidized to release energy to the body. So we say provide living organisms. Provide living organisms with nutrients that are oxidized to release energy to the body. That is an importance. Then we have excretion. Excretion, we know it's the separation and elimination of nitrogenous waste materials. Separation and elimination of nitrogenous waste materials from the organisms' cells or organisms' body cells. So, how does this characteristic help the organism? So, the importance is that it separates and removes waste products of metabolism, preventing cell poisoning. Separates and eliminates waste products of metabolism preventing cell poisoning. And this may even lead to the death of the whole organism. So, stating the importance correctly, you were supposed to be awarded 1 mark in each case. 1 mark in each case. Let's move to question number part B of this question. State the function of the following parts of a microscope. We have several parts of a light microscope, and one of them includes the objective lens. We have the parts of a microscope, and some of these parts include the objective lenses. And then we have the stage, and then we have a mirror. We have the diaphragm and the condenser. Then we have the coarse adjustment knob and the fine adjustment knob. We have the body tube. We have the eyepiece. We have the base. We have the stage, which carries the clips. We have the condenser, and at the lower side we have the diaphragm. Then we have the mirror. Then we have the arm. And then we have the coarse we have the coarse adjustment knob. We have the fine adjustment knob. Now, with all these parts, you need to know and understand all the functions of this particular microscope, parts of a microscope. So we know objective lens. There are 3 of them, from the one with the lowest magnification power, the one with the highest magnification power. And these objective lens, together with the eyepiece lens, they contribute to the total magnification of the specimen. So the answer to the role of objective lens, it contributes to the magnification of the object. Contributes to the magnification of the object. When I say it contributes, it means that it does not work alone. It works together with the eyepiece lens to bring about the total magnification. So it contributes to the total magnification of the object. Then we have the diaphragm. Diaphragm, as I have just indicated it's if this is the stage, then we have the condenser, and then at the bottom here we have the part with a regulator here, and this part is what we call the diaphragm. This is the part that we call the diaphragm, and then we have this part that we call the condenser here. And then we have the stage with clips. So what is the role of the diaphragm? Diaphragm, as you can see, the regulator here is to regulate the amount of light that reaches or gets onto the stage through the condenser. So the work of the diaphragm, it regulates the amount of light passing through the condenser to illuminate the specimen. So it regulates the amount of light passing through the condenser to illuminate the specimen. To illuminate is just to allow light or to brighten the specimen, or to allow light to reach the specimen. So that is the work of the diaphragm. You can use it, regulates or it controls the amount of light passing through the condenser to illuminate the specimen. And you can see where the diaphragm is. We can also have the condenser. Condenser, from the name condenser, it concentrates light onto the stage. The work of condenser is to concentrate light onto the stage. Condenser, if it comes in the exam, you have to say it concentrates light. It's not to regulate. This one concentrates light onto the stage. That is the work of the condenser, concentrate light onto the stage. Let's now move to question number 5. State 3 environmental factors that affect the rate of stomatal transpiration. We need to understand, learners, that the process of transpiration, which is the process by which plants lose water to the atmosphere in form of water vapor when the temperatures increase. This process is affected by some factors which are categorized into two. We have the stomatal, we have the structural factors which affect the process, and we also have the environmental factors which affect the process. So in our question here, we have been taught to state 3 environmental factors. So there is need that we underline the key term here. Those factors that are from the environment that affect the process of the rate of stomatal transpiration. So, what are some of these environmental factors? There are several. We are only taught to state 3. So we can talk of light intensity. Light intensity is one of the factor. We can also talk of temperature. We can also talk of wind, or the strength of wind. We have wind. We can also talk of atmospheric pressure. We can even talk of humidity and even water availability. Stating any 3, you were to score 3 marks. Remember, we also have the structural factors which affect the rate of transpiration, and this is where you talk of airy lips, thick cuticle, sunken stomata, the number of stomatas. Those are structural factors which affect the rate of stomatal transpiration. So always get to underline the key term so that you not be in a position to answer the wrong question. We don't fail because we do not know, we fail because of these minor mistakes that we do make. Let's move to question number 6, part A. What is the function of the following parts of the male reproductive system? We know under reproduction in Form 3, you learned the female reproductive system and the male reproductive system and their roles and the roles of those parts in the whole system. And some of the parts that you went through was epididymis in male reproductive system and the seminal vesicle and other accessory glands, including the prostate gland and corpus gland in the male reproductive system. So let's look at the function of these parts in the male reproductive system. Epididymis. Epididymis in the male reproductive system, we have the testes. No one is testis, and many are testes. Inside the testes, we have this coiled structure that we call epididymis. Epididymis is a structure for storing sperms. So we say stores sperms. It stores sperms. That is the function of the epididymis. Then we have the seminal vesicle. Is one of the accessory glands of the male reproductive system, including the prostate gland and the corpus gland. The seminal vesicle secretes an alkaline fluid which nourishes the spermatozoa. It secretes an alkaline fluid which contains nutrients to nourish the spermatozoa. So we say it secretes an alkaline fluid which contains nutrients to nourish the spermatozoa. The spermatozoa. As I have just said, we also have other accessory glands like the corpus gland, which also secretes an alkaline fluid that neutralizes the acidic urine in the urethra. It will also produce that alkaline fluid to lubricate the urethra to allow easier passage of the sperm. So this one, the corpus gland, this one secretes an alkaline fluid to neutralize acidic urethra. So the urethra of the male, to neutralize the acidic urethra. Then we have the prostate gland, which secretes an alkaline fluid that forms part of the semen, and it will also neutralize the acidic environment of the urethra and the female reproductive tract. So it will also neutralize the acidic environment in the female reproductive tract or the birth canal. So that is what we call the prostate gland. So prostate gland neutralizes the acidic environment prostate gland. So this one neutralizes the acidity in the female reproductive. Acidity in female reproductive tract. That is, it will also produce an alkaline fluid. These substances that are produced by these accessory glands, including the seminal vesicle, the corpus gland, the prostate gland, those substances that they produce, the secretions, together with the sperm, they form what we call semen. They form what we call semen. So the combination of all those secretions together with sperms, they form semen. Another part is the interstitial cells. Interstitial cells, they are also found within the testes, and these cells, they are there to produce the male sex hormones, the testosterone. So there are also special units of cells that produce the male sex hormone, the testosterone, which stimulates the production and the development of male secondary characteristics. So we can say the interstitial cells, they produce the male sex hormone, testosterone, produces or secretes male sex hormone, which is testosterone. This testosterone leads to stimulates sperm production, which stimulates sperm production, and also leads to development of secondary sexual characteristics in males, like deepening, breaking of the voice, broadening of the chest, and other characteristics that occur in males. So that is the work of interstitial cells. So stating these parts and their functions correctly, you are awarded 1 mark. Part B of the question. The diagram below shows the structure of a sperm cell. State the functions of the parts labeled A, B, and C. Let's begin by labeling these parts. We have A, which we call the acrosome. We have B, which we call the mitochondrion. Mitochondrion because it is one. So we have mitochondrion or mitochondrion. Mitochondrion is found in this part of the sperm that we call the middle piece. Middle piece. And then we have the tail. And we know in the mitochondrion, we have, in the middle piece, we have mitochondrion, it's packed with mitochondrion. And at one side, we also have centrioles for cell division and formation of spindle fibers. Now, let's give the functions of these parts of the male reproductive system of the sperm cell, that is of the sperm cell. A is acrosome. You know, acrosome, it contains lytic enzymes. Acrosome contains lytic enzyme, and this enzyme breaks the membranes of the ovum during fertilization. So we have those membranes of the ovum. They are broken by the lytic enzymes that are found in the acrosome before fertilization. So to pave the way for the sperm to enter into the nucleus of the ovum. So the role of part A, it contains lytic enzymes that dissolves the egg membranes during fertilization. We say contains lytic enzymes contains lytic enzymes that dissolves egg membranes during fertilization. We call them vitelline membrane during fertilization. Then we have part B. Part B, we have said this is a mitochondrion, and this mitochondrion provides the respiratory energy required for the propulsion of the sperm cell. So, provides respiratory energy required for the propulsion or movement required for the propulsion of sperm cell. We also have part C, which we say is the tail. The tail propels the sperm, the female reproductive system or tract. It propels the sperm in the female reproductive tract. So stating these points correctly, 3 marks, 1 mark each. Let's move to question number 7. Define the following terms. Define the following terms as used under the topic classification. What is a species? Species is one of the 7 taxonomic units that we talked about, or that you were taught in Form 3 or Form 1, Classification 1. So we have the 7 taxonomic units that I know you are given a way of memorizing them. That we say Kenya police can only fight German soldiers. And we have kingdom as the first taxonomic unit. Then we have the phylum in animals and division in plants. Then we have the class. Then we have order. Then we have family. Then we have genus. And we have what we are talking about right now, it is species. So species is the smallest taxonomic unit, or is the smallest unit of classification, whose members can freely interbreed to produce fertile offspring. And that's how we define a species. Species is the smallest unit of classification. Species is the smallest unit of classification, is the smallest unit of classification, whose members can freely or naturally interbreed to produce fertile offspring, or to produce viable offspring. So that's how we define a species. Species is the smallest unit of classification whose members can freely interbreed to produce fertile offspring. And as you can see, of course, it's the smallest unit of classification. And together with genus name, we use to give organisms scientific names using binomial nomenclature. So, what is binomial nomenclature? It's another double naming system of organisms. So, binomial nomenclature, in simple terms, we say is a system of naming organisms using two names. That's the genus name and the species name. So we say is a system of naming organisms using two names which are scientific. It's a system of naming organisms using two names, that is genus and species name. Genus name and species name. You may also find another person say is a double naming system of organisms using genus and species name. Remember, we also have the principles that guide the binomial nomenclature, how it's supposed to be done, how the names are supposed to be written. So stating this gives you 2 marks, each 1 mark. Let's move to part B of this question. A certain sea animal has a smooth skin, lungs, regulates its body temperature, and gives birth to young ones. To which class does the animal belong? So we know under the kingdom Animalia, we have 2 phylums that are learned or they are taught under the 844, that is between Form 1 and Form 4. And among these 2 phyla, we have the phylum Chordata. The phylum Chordata, whereby you are given or you gave several classes, including class Mammalia, class Reptilia, class Apes, class Species, class Reptilia, Amphibia, all those classes. And these classes, organisms have unique characteristics that you may not find in the other group. You may not find all the characteristics, the all characteristics they have in the other group of organisms. So when you look at the characteristics that you are given here, this organism is a sea animal with a smooth skin, have lungs, regulates the body temperature and gives birth. Regulating body temperature and having lungs. We have regulating body temperature and giving birth. Those 2 statements alone are enough to help us classify these organisms. And these characteristics are basically for a mammal. So the class is class Mammalia. So we say Mammalia, class Mammalia, and this gives you 1 mark. Reasons is because they are able to regulate their body temperature, they are able to give birth to young ones. I want us to move to question 8. I want to move to question 8. For a leaf to be efficient for photosynthesis, it has to be broad and flat. Explain. So here we have the leaf being broad and flat. The reason as to why a leaf has to be broad is one, to give the leaf a large surface area so that it absorbs more sunlight. And the reason as to why the leaf is flat is to allow more light to reach the photosynthetic cells, or what we call the chloroplast. So we say a broad leaf has large surface area for absorbing sunlight, while a flat shape allows maximum exposure to light. So we can simply say to allow or to provide large surface area for absorption of light, maximum amount of light. That is the reason as to why it is broad, and it is flat for more light to reach the photosynthetic cells, and flat for more light to reach the photosynthetic cells, which are the chloroplasts. So stating that 2 marks. Let's move to question number 9. In an accident, a victim suffered brain injury. Consequently, the heartbeat was affected. Name the part of the brain which was injured. In Form 4, the topic response, you learn parts of the central nervous system, including the brain. In some of the parts of the brain, we have the cerebellum, we have the medulla oblongata, we have the pons, and other parts such as the pituitary gland, hypothalamus, and other parts. Those parts have different functions and that they perform that lead to the control of activities that do take place in the body. Here, we are told this person was involved in an accident and they suffered brain injury. Consequently, it was noted that the heartbeat was affected. What is this that is connecting the brain with the heart? So that now the heart is not functioning the way it's supposed to be functioning. So then we are being told to name the part of the brain which was injured. So we have this part in the brain that we call the medulla oblongata, which controls the involuntary activities in the body, including heartbeat. Those activities that the body, you as a person, you don't need to decide whether they are supposed to occur or not. For instance, you don't decide for the heartbeat to continue. You do not decide whether to breathe or not. You don't decide the blood pressure. You also do not decide the process of swallowing and even coughing and sneezing. So those activities, we call them involuntary activities, and they are controlled by the medulla oblongata. So the answer here was medulla oblongata. Medulla oblongata. So this is the part which controls involuntary activities. involuntary activities, including the heartbeat. We have the cerebrum that controls memory and will also control the voluntary activities such as movement of skeletal muscles. So that part is different from medulla oblongata. So whatever the cerebrum does is opposite to what medulla oblongata does, because medulla controls the involuntary movements or activities, while cerebrum, it controls the voluntary activities in the body. So the correct answer to this question was medulla oblongata. I want us now to move to question number 10 of this particular paper. Question number 10 says, state the products of anaerobic respiration in animals. State the products of anaerobic respiration in animals. In the previous question, I've just talked about the process of respiration, the phases of respiration under the anaerobic or aerobic respiration. And I've said that under the phase glycolysis, glucose is broken down to pyruvic acid first, and then it is converted in plants into ethanol, and in animals to lactic acid in absence of enough oxygen. So we are being told, say, the products of anaerobic respiration in animals. So the products, here we have the major products, we have lactic acid and energy are the major products of anaerobic respiration. So we have lactic acid, and we also have energy as the products of anaerobic respiration in animals. In plants, the products of anaerobic respiration is ethanol, water, and carbon dioxide. And those products, because of carbon dioxide, we have talked about ethanol. So that is how fermentation occurs. And we have several applications of the process of anaerobic respiration in plants. So these are the products in animals. So in plants is ethanol, carbon dioxide, and water. But in animals, we have lactic acid, which we say it accumulates in the body muscles, and then we have the energy. Let's now move to next question. Next question says, state 2 physiological changes that take place in human skin in order to facilitate heat loss from the body. State 2 physiological changes that take place in the human skin in order to facilitate heat loss. Skin is one of the organs that bring about thermoregulation. Is an homeostatic organ, skin. It regulates body temperature. And this particular organ, it works, it has certain structures or certain parts or organs that make the process of thermoregulation a success. These organs in the skin that bring about thermoregulation, we have blood vessels. We also have the sweat glands. We also have the hair and the erector pili muscle. The erector pili muscle. So these parts work independently to bring about the issue of thermoregulation. But now we are being told to state two physiological changes that take place in human skin in order to facilitate this heat loss from the body. So some of the physiological processes, because we have the sweat glands producing more sweat, so we can talk of sweating or secretion of sweat as one of the physiological process. We can talk of sweating, or we can say secretion or production of sweat. We can also talk about what happens to the blood vessels because we are losing heat. So the blood vessels vasodilate. So we have vasodilation. So that blood flows near the skin surface. So we have vasodilation. We also have the erector pili muscle relaxing and the air lying flat. So we have relaxation of erector pili muscle and the air lies flat on the skin surface. So those are some of the physiological activities that take place on or in the skin, human skin, to facilitate heat loss from the body. So any two was enough to give you a total of 2 marks. But B of this particular question, state one function of sebum. State one function of sebum. When you are looking on the structure of the mammalian skin, one of the parts that you looked at was the sebaceous gland. The sebaceous gland. Sebaceous gland is the gland that secretes sebum. And sebum has two major functions. And one of the functions, it makes the skin to supple. It makes the skin to supple, or it moistens the skin. Makes the skin to supple. You can also say it moistens the skin. Another function, it is antiseptic. It is an antiseptic. It is an antiseptic. Antiseptic means it has medicinal properties. It means it has medicinal properties. So antiseptic spellings very crucial. It is an antiseptic. So it has medicinal properties, or you can still say it kills pathogens or germs on the skin. Please don't say it acts as an antiseptic. It does not act, that is function. It is an antiseptic, that is role. It does not act. It carries its role fully. It does not act. It is an antiseptic, or you can say it kills germs. One function, 1 mark. Let's move to the next question. Part 12. Define the term homeostasis. Define the term homeostasis. Homeostasis is one of the subtopics that we learn together with excretion. And homeostasis, we refer to it as is the maintenance of constant internal environment despite changes in the external environment. So that is what we call homeostasis, is the maintenance of constant internal environment despite changes in the external environment. So that is what we call homeostasis. So for instance, even I have an homeostatic factor such as temperature. In mammals, temperature is supposed to be maintained at around 36 degrees Celsius. So despite temperatures increasing outside the body or outside the environment which surrounds the cells, the temperature within the body cells will remain to be 36. Even if the external temperatures reach or get to 40 degrees Celsius, the internal temperature of a mammal will remain, or a human being will remain at 36 degrees Celsius. So that is a factor which is maintained at constant. Other factors which are maintained at constant, we have glucose levels in the blood. We also have the ions in the body, water, and salts. Those are some of the homeostatic factors which are maintained at constant in living organisms. Let's move to the next question, or part B of this question. State one way by which mammalian blood carry out homeostasis function. Carry out one way in which mammalian blood carry out homeostatic function. So one of the functions, one way in which mammalian blood carry out homeostatic function, we can talk of blood transport hormones and other substances around the body, and this helps to regulate body processes and maintain a constant internal environment. So we can say blood transports hormones and other substances around the body, and other substances around the body, and help to regulate processes and maintain a constant internal environment. For instance, we know very well that for glucose to be maintained at constant, we need two hormones. We have insulin and we also have glucagon. So these glucagons are produced in the pancreas, and they have to be transported to the liver to function. So if it were not for this blood to transport these hormones, then maintenance of this particular factor in the body, it could have been a problem. So we see blood transporting hormones that later leads to regulation of these particular factors or processes in the body. We can also talk about blood regulating the body temperature by transporting it. They distribute it within the body. So blood itself, it can help to maintain a constant body temperature, especially in mammals. And stating that gives you 1 mark. Let's move to question number 13. The diagram below represents certain stages of cell division. Study them and answer the questions that follow. We have R, S, and T. Looking at types of cell division, we have two major types of cell division. We have mitosis and we have meiosis. Mitosis is a type of cell division that occurs in somatic cells or the body cells, and it only involves one process or one type of cell division, one cell division. Then you have meiosis, which takes place in the reproductive cells, like in the testes and also in the ovary, and it leads to formation of gametes. It involves two divisions, two meiotic divisions, and under them we have the phases of cell division under mitosis and also in meiosis. Like I said, we always use acronym IPMAT. IPMAT, I standing for interface. We have prophase. We have metaphase. We have anaphase. And we have telophase. Not forgetting that in meiosis, we have meiosis 1 and meiosis 2. So it means that we have the cell division 1, we have meiosis, the first meiotic cell division and the second meiotic cell division. So in first meiotic cell division, we are going to use terms like interphase 1, prophase 1, metaphase 1, anaphase 1, and telophase 1. In second meiotic cell division, we are going to use terms such as interphase 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. So according to what you understood in that particular topic, we have the diagrammatic representation of what happens during those various stages of phases of cell division. So when you look at R, you can notice something, that the chromosomes are in pairs. And these chromosomes have been arranged at the equator of the cell. At the equator of the cell. And when you look at them, the chromosomes are homologous pairs. They are in pairs. Basically, the phase which has this kind of characteristics is metaphase 1. Because chromosomes are arranged at the equator and they are in pairs. So this is metaphase 1. And then we have S. When you look at it, it's not very much different from the metaphase. There is only constriction of the cell trying to divide, and there is separation that the chromosomes, the homologous chromosomes, are trying to separate to move to the opposite poles. And when you look at this, automatically it becomes the phase that comes immediately after the metaphase. And the chromosomes, remember, are still in pairs, but they have separated. So the homologous chromosomes have separated. So S becomes anaphase 1. This was metaphase 1, and S now is anaphase 1. S is anaphase 1. Anaphase 1. Then we have T. When you look at T, these are two independent cells which are trying to divide. So this is a cell by itself, and this is another cell. And when you look at them, they are dividing. We have this constriction that is occurring in each respective cell. So it means that the S and T, they are in the same stage. But now, T is leading to formation of 4 daughter cells, unlike S, which is trying to form two daughter cells. So T is anaphase, but it's anaphase 2. T is anaphase, but anaphase 2, because the chromosomes are not in pairs. We already have chromatids in each of the four daughter cells. So this is anaphase, but anaphase 2. So confidently, after that analysis, we can name the phases of cell division represented by the diagrams R and S. They have given us R, S, and T. So we are supposed to have also T at this point. So R is metaphase 1. Metaphase 1. S is anaphase 1. And T is anaphase 2. Question number 14. State the functions of the following organelles. We have several organelles as observed under the plant cell and animal cell, which they observed under the electron microscope. And one of the organelles is the lysosome. The main function of lysosomes, they destroy, or they contain lytic, or they have lytic enzymes that destroy worn out organelles, cells, or tissues, or the entire cell. So they have, or they produce or secrete lytic enzymes that destroy worn out organelles, tissues, or the entire cell, They may also be involved in the process of digestion of pathogens and bacteria, and they will be found in phagocytes. They can also be involved in digestion of food materials. Then we have the Golgi apparatus. Golgi apparatus, they help in packaging and transportation of glycoproteins. Helps in packaging and transportation of glycoproteins. You can also find them being used in packaging of synthesized materials and transporting them. We can also talk about helps in production of lysosomes. It can also be involved in the production of synthesis of lysosomes that we have just talked about there up. So each statement or each function here was giving you a total of 1 mark. Moving to question number 15. Explain why the carrying capacity of wild animals is higher than that of cattle in a given area. Explain why the carrying capacity of wild animals is higher than that of cattle in a given area. We know carrying capacity is the maximum number of organisms an area can comfortably support without the depletion of the available resources. And these resources may include food and water. So, why is it that wild animals, they are accommodated? A lot of wild animals, or very many wild animals, are accommodated in a given area as compared to cattle. So we need to understand that in this case, wild animals, most of them, they are both browsers and grazers. So they are browsers and grazers. So they are able to exploit such habitats with leaves and grass at the same time. But the cattle, they are only grazers. So when it comes to competition, there's a lot of competition when it comes to cattle because they are only depending on grass, and therefore that particular given area will only accommodate the required very small number or a less number or lower number of organisms or cattle as compared to wild animals. Because in the same given area, they cannot consume the cattle, cannot consume the leaves, but they can consume the grass. So we say wild animals. We can write and say what animals have different feeding habits. So wild animals have different feeding habits, which include they are browsers and grazers, and therefore they are browsers and grazers, hence minimum competition. Hence minimum competition. Hence minimum competition for food. While cattle have one feeding habit, while cattle have one feeding habit. In other words, they are grazers only, and hence more competition, for available grass. Hence fewer number of cattle supported in a given area. Supported in a given area. So, giving that description 3 marks. 3 marks. Question number 16. Study the following diagram and answer the questions that follow. Look at these organisms. You notice that these are the members from the phylum Arthropoda under the kingdom Animalia. And we know under the phylum Arthropoda, we have several classes, which includes class Insecta. We have several classes, which includes class Insecta. We have class Chilopoda. We have class Diplopoda. We have class Crustacea. We have class Arachnida. So, among these organisms that we are given in these particular diagrams, they belong to one of these classes or two of these classes. So, as you can see, from the knowledge of observing organisms in our environment, A is a millipede. Millipede, we said, has 2 pairs of legs per segment, and as you can see, it has a round body or a tubular body, and then we have the centipede. the centipede, which is dorsoventrally flattened, and it has one pair of legs per segment. So we say that millipede is classified under the class Diplopoda because of two pairs of legs per segment. Diplopoda. And we have B, which is centipede, which lies under the class Chilopoda. So we can comfortably answer the question, to which class does each of the specimen belong? A is class Diplopoda and B is class Chilopoda. Each 1 mark, total 2 marks. Part C, name 2 classes of phylum Arthropoda with a cephalothorax. Cephalothorax, it is where the head and thorax are fused to form one unit, and this only occurs in classes Arachnida and classes Crustacea. So we have Crustacea and Arachnida with a cephalothorax. So we have class Arachnida and we have class Crustacea. So each 1 mark. Question number 16. Question number 16. State one reason why genetists prefer to use Drosophila melanogaster, which is the fruit fly, for genetic experiments. Scientists used Drosophila melanogaster in the study of inheritance, especially Thomas Morgan used this particular fruit fly, an insect, during his research in the study of inheritance. Some of the reasons which led to these scientists to use this method was Drosophila melanogaster breeds so fast. So we can say it breeds so fast. It breeds so fast. You can also say it produces many offsprings. Produces many offsprings. We can also talk of it as a short life cycle. As a short life cycle. So any one of these statements you were to get 1 mark. We can also talk of it is harmless and it's easy to handle as another reason why this scientist by the name Thomas Morgan chose Drosophila melanogaster. Moving to next question number 17. What's the significance of active transport in human body? Significance is the importance of. So active transport is where materials move against the concentration gradient by use of energy. And in this case, active transport can be used in absorption. It helps in absorption of digested food materials from the digestive tract to the bloodstream. You can also talk about in the reabsorption of useful materials in the kidney nephron. In the kidney nephron. We can also talk of it helping in the pumping mechanism of sodium ions in the nervous system. So any of these two statements, you'll get 2 marks. Moving to question number 18, in an attempt to clear certain weed from Ujugora fish pond, St. Matthew's Secondary School biology students introduced a species of beetles into the pond. Give the term that we use to describe this method of control. The method where we use a living organism to eradicate another living organism is what we call biological control method. This method, we call it biological control. So the answer is biological control. Question number 19. State 2 advantages of this method over the use of herbicides. This method is harmless to non-target organisms. Remember the use of herbicides. Herbicides are chemicals, and therefore it may lead to death of non-target organisms. So one of the reasons for advantages is that non-target organisms are never killed. We say non-target organisms are never killed. Another reason or advantage is that there is less or no chances of water pollution. There are no or less chances of water pollution. So these two statements, 2 marks. Question number 20. State the causative agent of cholera. Cholera is one of the bacterial disease that is waterborne. And the causative agent of cholera, we call it Vibrio cholerae. And we use the principles and the rules of binomial nomenclature to give the name of the causative agent. Therefore, we write Vibrio cholerae. We underline the name separately simply because it is unwritten. We also have typhoid, which is another bacterial disease. The causative agent is Salmonella typhi. Again, you follow the rules of writing any scientific name to name the causative agent. What is the cause of the hydration in cholera victims? In this case, the hydration is excessive loss of water from the body, and this results due to excessive diarrhea or vomiting. So you can talk about the cause is excessive diarrhea. Excessive diarrhea is the main cause of dehydration. Question number 21. The numbers of chromosomes in a gorilla chick cell is 48. State the number of chromosomes in a gorilla's ovum. We know that gorilla chick cell is part of the somatic cells. Somatic cells of an organism has twice the number of chromosomes that the reproductive cells should have. So if the gorilla cell has 48 chromosomes, the somatic cells, we are being asked, what about the number of chromosomes in the gorilla's ovum? Gorilla ovum, this is a reproductive cell. Therefore, it should be half the number of the chromosomes in the somatic cell. Therefore, the number of chromosomes is supposed to be 24 chromosomes. This is because a reproductive cell is haploid while a somatic cell is always diploid in nature. Part B of this question, state the role of each of the following during cell division. Centrioles during cell division, we are able to identify the position of the centrioles. The work of the centrioles is to form spindle fibers. So they involved in the process of spindle fiber formation. So forms spindle fibers. Then we have the question on the function of the spindle fibers. These spindle fibers, after they are formed by the centrioles, it is where the chromosomes attach using the centromere and they facilitate cell division. So attached to the centromere of the chromosomes, attached to the centromere of the chromosomes to facilitate cell division. Again, each 1 mark. Moving to the next question, number 22. The diagram below shows a germinating seedling. Name the part of the seedling labeled B. We know we have two major types of germination. We have epigene germination and we have hypogene germination. Epigene germination is a type of germination that occurs in dicots. The cotyledons, they are brought above the earth's surface. The cotyledons are brought above the earth's surface, while hypogene, the cotyledons are left underground. And during germination, in epigene germination, we have, like in the epigene germination, like in the case of bean germinating, we can see the presence of a taproot. We can also see the cotyledons. It is a cotyledon. In between the cotyledon and the taproot, we have the part that curves to force the ground to break so that the cotyledons are brought above the soil surface. And that part is what we call hypocotyle. Hypocotyle. So you notice that the type of germination in dicots is epigene germination, and they have a part known as hypocotyle that may lead to confusion because the type of germination in monocots is hypogene and they do not have hypocotyle. Hypocotyle is a part in dicots under the epigene type of germination. If I have a main seedling germinating, we can see the fibrous roots and the cotyledon being left underground. They have a part, a section that we call epicotyle. Remember, the type of germination is hypogene. So these two terms, they may bring confusion. We have germination in monocots, hypogene. They have a part known as epicotyle. Germination in dicots, epigene. They have a part known as hypocotyle. So they name the part of the seedling labeled B because this is epigene germination. You can see this is a bit. You can see the cotyledons being brought above the earth's surface. And part B becomes the hypocotyle. Hypocotyle. 1 mark. State the type of germination exhibited above. As we have just seen, this is epigene germination. Epigene germination. 1 mark. Question number 23. Name 2 hormones that control metamorphosis in insects. In Form 3, growth and development, talked about hormones, role of hormones in metamorphosis. And we have 2 major hormones in insects that regulate the process of metamorphosis. And one of them is juvenile hormone, which controls or brings about formation of larval cuticle. We also have ecdysone. Ecdysone, also known as molting hormone, which brings about molting out of the adult cuticle or exoskeleton. Therefore, stating this 1 mark each. Question number 23. We have 223. It's a typing error. What is the importance of the following plant excretory products? We have several excretory products under excretion in plants. These plants excrete some products that are useful. In some of the products we have papain, nicotine, quinine, as they have been indicated. We also have cat or mira, heroin, cocaine, and many other excretory products in plants that are useful. The function of papain, which is extracted from a pawpaw plant, it's used as a meat tenderizer. Used as a meat tenderizer. We have nicotine, which is used to manufacture insecticides, and again, it's used as a narcotic drug. Used to manufacture insecticides, and can also be used as a narcotic drug. Quinine, which is commonly tested in exams and is common, we interact with some of the plants that contain it, like the aloe vera. It's used for treatment of malaria. Used in treatment of malaria. Question number 24. So these statements each 1 mark. Question number 24. Name the carbohydrate that is found in abundance in mammalian blood. Initially or previously I had talked about types of carbohydrates, and I said that carbohydrates, they have 3 categories. We have the monosaccharides, the disaccharides, and the polysaccharides. The monosaccharides, we have glucose, we have fructose, and we also have galactose. In disaccharides, we have maltose, which is formed from combination of 2 glucose molecules. We also have sucrose, which is formed from glucose and fructose, and we have lactose, which is from galactose and glucose. We have examples of polysaccharides like starch in plant tissues. We have glycogen in animal tissue. We also have chitin or chitin in exoskeleton of arthropods. We have lignin as found in xylem tissues and xylem tracheids, xylem vessels and xylem tracheids. And we also have cellulose, which is the type of carbohydrate that's found in, or the tissue that forms the cell wall in plants. Glucose, which is a carbohydrate under monosaccharides, it's abundant in mammalian blood. Fructose is abundant in ripe fruits. Galactose is abundant in milk. Maltose, sucrose, and lactose, because they are products of monosaccharides, we will not mention where they are abundant in. Polysaccharides we have starch abundant in plant tissues. Glycogen is abundant in animal tissues. We also have chitin, which is abundant in exoskeleton, lignin abundant in xylem, and also sclerenchyma tissue, and cellulose abundant in plant cell wall. So answering this question, we name the carbohydrate. All these that I've mentioned, they are all referred to as carbohydrates. So we know the categorization of monosaccharides, disaccharides, and polysaccharides. They are all of them, they are carbohydrates. Be it glucose, be it fructose, be it maltose, be it sucrose, be it starch, all those are carbohydrates. Therefore, the carbohydrate that is abundant in mammalian blood, we have said, is glucose. We also have the carbohydrate that's abundant in mammalian liver, that I've said that in animal tissue. is glycogen. Glycogen is the storage form of glucose. So once glucose is in excess, is converted to glycogen, then stored in animal tissue. Therefore, again, you will be awarded 1 mark. If we had stored in abundance in ripe fruits, then it could be fructose. If it were for milk, galactose. If it were for plant tissues like seeds, starch. If it were for exoskeleton, chitin. If it were for xylem tissues, lignin. If it were for cell wall, then it could have been cellulose. So let's move to question number 25. Name 2 tissues in plants that are thickened by lignin. We have 2 main tissues. We have several tissues in plants, including sclerenchyma tissues, including phloem, xylem. But the only tissues that are thickened with lignin, we have sclerenchyma tissues, sclerenchyma tissue, and we also have xylem vessels and xylem tracheates. They are thickened with lignin, each 1 mark. Question number 26 states the role of muscles in the stomach during digestion. We have the circular and longitudinal muscles that are involved in the process of mixing food or the contents in the stomach when they contract and relax. So they contract and relax. These muscles contracts and these muscles contracts and relaxes to mix the contents of the stomach. So 1 mark. Name the antigens that determine human blood groups. We have 3 major antigens that determine blood groups in human beings. We have the obvious ones. We have antigen A and we also have antigen B. We do not have antigen O. We have O is a blood group. They do not members with blood group O, they do not have antigens, and therefore the only antigens that we have is A and B. And we also have the rhesus antigen or the rhesus factor. The rhesus factor, also known as antigen D. So this gives you 2 marks. Moving to the next question, number 28. State 3 methods of fossil formation. State 3 methods of fossil formation. We have several methods of fossil formation. I'm going to list them down. We have petrification as one of the method of fossil formation, as explained under the evidences of evolution. Petrification is whereby the remains of an organism are gradually replaced by minerals from the surrounding water. So, for example, you find a bone changing to stone-like substance. That's petrification. We also have replacement. Replacement is whereby this is where the original material of the organism is dissolved and replaced by a mineral. For example, a wood is replaced by minerals and it becomes fossilized, or it becomes a fossil. Then we have carbonization. Carbonization. This is where soft parts of an organism are compressed and most of the substances that are in that organism are removed, and they leave a thin layer of carbon, and that's what we call carbonization. We also have mold formation and cast formation. We have mold formation and we also have cast formation. So mold formation is whereby an organism is buried and later it decays. When it decays or dissolves, it leaves its shape or what we call an impression in the rock or where it has just dissolved. So I have a picture of having a duff of chapati, cooking chapati, and then you force your wrist or fist through it. So what will happen? You are going to leave an impression of that. So what happen? That is what we call the mold formation. Then you have the cast formation. If that mold that has been left there is filled with materials, and then those materials later hardens, so that when I remove that material, it exactly looks like your wrist, then that is cast. So we also have cast formation. So those are some of the methods of fossil formation. Any two, any three, you are to get 3 marks. Question number 29. Name the type of tool that is used by a dog to slice flesh from the bones. We have dogs which are carnivores, and we know carnivores, they have special type of teeth that we call, apart from the canines, we have the tooth that we call carnassial tooth or carnassial teeth. This carnassial tooth is specialized for seizing and slicing flesh from bones. So in this case, the name of tooth is what we call carnassial tooth. Explain 2 ways in which the tooth is adapted to its function. This tooth has sharp edges with smooth sides to slice flesh. Has sharp edges with smooth sides to slice flesh. We also have another adaptation. They have cusps and ridges. Have cusps and ridges to increase surface area for crushing bones. So these statements will give you 2 marks. Question number 30. The diagram below represents a portion of a certain nucleic acid. We have 2 major types of nucleic acids. We have the ribonucleic acid and we have the deoxyribonucleic acid. The first one is RNA and the second one is DNA. RNA has a ribose sugar and DNA has a deoxyribose sugar. The major difference between the two in terms of their structure, DNA has base thymine. DNA has base thymine, which is replaced by base uracil in RNA, which is replaced by base uracil in RNA. So looking at our questions with a reason, identify the type of nucleic acid shown above. So this type of nucleic acid is a ribonucleic acid, or what we basically call RNA. Give a reason for your answer. because of the presence of base uracil. Because of presence of base uracil. So these statements, 1 mark each. Part B of this question, name the part of the cell where the section of the nucleic acid is shown would be used. So we know in a cell, the genetic materials are found or they are formed in the what we call nucleolus. Once they are formed in the nucleus or nucleolus, they are used in the cytoplasm. That is where they are used because they transport. We have the formation of proteins. So they are used in the cytoplasm. So the correct answer is cytoplasm. So this marks the end of our presentation. You found this video useful to you. Please don't forget to subscribe to our YouTube channel, Training Grades Online School. Share our video to reach as many students as possible who may require our assistance. And most importantly, comment your reactions and your views concerning this video and also concerning the type or the topic that you'd like us to cover or the paper that you'd like us to cover in our next presentation. And again, subscribe to our YouTube channel, hit the subscription button, the notification button, so that you be notified whenever we drop a video of this kind. Thank you so much for watching, and thank you for your time. May God bless you and have a nice and wonderful time, and all the best in your upcoming examination. May you pass with flying colors. Thank you.